Then next one. Which one of the following options contain only those type of mapping population that are characterized by true breeding individuals? So true breeding line is produced by continuous self-pollination, and this continuous self-pollination, it increases the homozygosity in the individual. That is true breeding. So RILs, recombinant inbred lines. So it is formed by crossing two inbred strains followed by repeated self-pollination to create new inbred lines whose genome is the same as that of the parental genome. So it is a true breeding. So they are homozygous lines and they are true breeding. But F2 populations are not. Then second option, if you see F2 population and BC1, BC is backcross 1 F2. So that is also not correct. Double haploid population. Yes, double haploid population are also homozygous lines. And they are obtained by chromosome doubling of haploid plants, which are derived, that is, these haploid plants are derived from anther or pollen culture, and then these are homozygous lines, double haploid populations. So if you see option 4, double haploid population and recombinant inbred lines, so both are correct. So answer is option 4. Then next one. In a family, the father has an X-linked mutation causing a late onset lethal disorder, and the mother is not a carrier. Mother is not a carrier of the mutation. Based on the above information, which of the following statements about the children the couple may have is correct? So this is the father, this is the disease carrying gene XD and Y, and this is mother. These are the gametes. Mother is not a carrier, so she is totally normal. The daughter will inherit one X from the father and one X from the mother. The son will inherit the X from the mother and Y from the father. So there is 50% chance that son will show the disorder. No, there is no chance that son will show the disorder. No children will show the disorder. You cannot completely say that. The probability that the parent have a daughter carrying a mutant allele is 25%. It is not 25%, it is 100%. The daughter will carry a mutant. See here, only daughter and son. So the daughter carrying the disease is 100%. All daughters will carry the mutant allele. That is correct. So the answer here is option 4. Then next one. In a mutagenesis experiment, the following pedigree was obtained. All progeny had the same phenotype. The mutation is most likely to have occurred. So here the mother nor the father is affected, but all the daughters and all the sons are affected. So this is a case of maternal effect of the genes. Here the genotype of the mother determines the phenotype of the offspring. And the gene products from maternal effector genes such as RNA and proteins, they are made during oogenesis in the mother's germ cells. So here this is an example of maternal effect genes. The mutation is most likely to have occurred during maturation of the egg cell, not during maturation, before that. In the maternal somatic cells, if it was maternal somatic cell, then mother should have shown the disorder. But here it is not the case. In the mother, in the precursor to all germ cells, yes, that is correct. In the embryo during development, that is also not correct. So answer here is option 3. Then, during animal cell culture, which one of the following cell types would not require trypsin for passaging? So, when you add trypsin to cell culture, what it does is it will break down the proteins that enable the cell to adhere to the vessel. Okay. So if you so you have to see for non-adherent cells from the options. So it is hematopoietic cells that are non-adherent, others are all adherent cells. So the answer here is option 2. Then next one. In the production of yeast alcohol by fermentation in the absence of oxygen, yeast convert glucose to pyruvate and pyruvate to ethanol. Fermentation promotes glycolysis by. So when glucose is converted to pyruvate, NAD plus is converted to NADH. The lost NAD plus is regenerated through citric acid cycle and electron transport chain under aerobic conditions. But in anaerobic condition, it is not so. There is no citric acid cycle and electron transport chain happening. So NAD plus will not be regenerated. So what the bacteria does is for regeneration, yeast does is for regeneration of NAD plus so that glycolysis will keep occurring. What it has to do is it has to first convert pyruvate to acetaldehyde and then acetaldehyde to ethanol. And this will result in the conversion of NADH to NAD plus, which will replenish glycolysis. Okay. So what does how fermentation promotes glycolysis in anaerobic condition is by generating NAD plus for glycolysis. Answer is option 4. Then next one. Which one of the following is not required in the blood glucose biosensor? So a biosensor, it is a device that uses living organisms or biological molecules such as enzymes and antibodies to detect the presence of chemicals, and in glucose biosensor they detect the presence of glucose and it is involved in managing diabetes. So the common enzyme that is used to detect glucose in the glucose biosensors are glucose oxidase and glucose dehydrogenase. Then it has got, this is your biosample. This is the biosample that has got glucose in it, or your blood sample that is recognized by the bioreceptor. Bioreceptor is nothing but the enzyme that will recognize the glucose and bind to it. So here it is glucose oxidase or glucose dehydrogenase. Then there is the transducer. Transducer, it is a part of the biosensor that converts one form of energy into another. Then the next is signal processing and display. And these signal processing components, they will process the transduced signal and prepare it for display. So that is what is taking place in biosensor. So glucose oxidase is biologically active component. That's true. They have asked which of the following is not required in glucose biosensor. Electrode as electrochemical transducer. That's also correct. Piezoelectric crystals as sensor. That is not correct. FAD as coenzyme of glucose oxidase. Yes, you can have coenzymes like FAD and NAD as well as PQQ. PQQ is pyrroloquinoline quinone. So these are the coenzymes that are involved in biosensor enzymes of glucose biosensor. Then next one. Which of the following combinations of excitation, light and objectives give the best lateral resolution in a fluorescence microscope? So whatever be the microscope, the resolution, it always depends on lambda and numerical aperture. So for higher resolution, you have to decrease the lambda or increase the numerical aperture. So decrease the lambda, you can see in two options, that is 1 and 4. These 2 and 3 can be ruled out. So 1 is 480 and 4 also 480 nanometers. Next is you have to see for the numerical aperture, which is which should be higher for higher resolution. So out of these two, the numerical aperture 1.1 Na and 1.4 Na, 1.4 Na is higher. So the answer here is first one, and 63x or 100x, it doesn't have anything to do with resolution. It is just magnification. So the answer here is option 1, 480 nanometers, 63x and 1.4 Na. Then next one. Which one of the following techniques allow the study of all three types of interaction, namely protein-protein, protein-DNA and protein-RNA? So it is phage display that is involved here. So phage display technique, the gene encoding a protein of interest, it is inserted into the phage coat protein gene, causing the phage to display the protein on the outside. That is called as phage display. That is, you insert your gene of interest into the phage coat protein gene so that phage will display it on the outside. And the main advantage of this methodology is that there is a direct link between the phenotype and the genotype. So if you are adding gene A and you want to see where it will express, and this gene A, it will express on the outside of the phage, that is on its coat protein. So once you see this particular protein there, you can conclude that gene A is present in the genome. So there is a direct link between the genotype and the phenotype in phage display. So how do you create a phage display cycle? So here you can see this is a population of Fab genes. Fab genes are found in your antibodies. So this is a phage Fab library. So different type of Fab genes are there. So if it is brown color, phage will express brown colored protein on the outside. If it is black color, it will express black color protein on the outside. If it is green color, it will express green colored protein on the outside. Now all these phages, they constitute what is called a phage library of Fab genes. Now these are incubated with immobilized antigen. Now antigen, if it is a protein, then it is protein-protein interaction. Fab genes, antibodies, so protein-protein interaction. Then what you do, the next step is unbound phage. Now when you subject it to antigen, only those Fab genes which are of interest to you, that is, they will bind to this antigen. Or only those Fab gene proteins that are complementary to this antigen will bind there. And this will unbound phages are then removed by washing. Then the good ones which have bound to the antigen are eluted. After eluting, these phages are subjected to infection to E. coli or bacteria. Then amplification will take place there, and the process is repeated 2 to 3 times, resulting in enriched population of antibody or peptide fragment or any antigen of your interest. Then you can screen for these phages, whether it is the same phage or the Fab gene that you have taken as your gene of interest. Now if it is protein DNA interaction, here in this case the phage library of transcription factors will be used, and here instead of antigen it will be various DNA molecules. Then you repeat the same process and you will get your come to know whether this protein is binding with this particular DNA or not. So to study protein DNA interaction or protein RNA interaction, phage display is used. So the answer here is option 2. Then next one. What type of electromagnetic radiation is used in biomolecular NMR spectroscopy? So it is radio waves that are used. Answer is option 1. Which one of the following statistical methods compares the means of the population? So it is t-test and analysis of variance. They both compare the means of the population. Then which one of the following enzyme does not catalyze the oxidation of substrate by reducing the electron acceptor NAD+? So here if you see the option succinate dehydrogenase, it is complex 2 and it does not catalyze, it does not reduce the electron acceptor NAD+, but instead it will reduce FAD to FADH2. So the answer here is option 3, succinate dehydrogenase. So that's it about part B. Part C I will be doing. Thank you for watching my channel. Please do like, share and subscribe. Thank you.